Hey everyone, welcome back to my class. In today's section, we are going to study about the second part of the chapter, that is locomotion and reproduction. So second part is your reproduction. Let's begin our class. What is reproduction? Reproduction is nothing but the formation of new daughter cell is formed of a similar kind. Like human is going to produce human, monkey will produce monkey, bacteria will produce bacteria. Have you ever seen human will produce bacteria or bacteria will produce human? No, they are going to produce the similar kind of new individual. This process we call it as reproduction. What is the purpose of reproduction? Why do we do it? What is the importance of it? It is for the continuity of species. Now suppose if a species is not doing reproduction, so if it doesn't do reproduction, after its lifespan what will happen? All the species will die and there will be extinction of the species, right? Now it is not required for the survival of individual. Can I say that if I won't do reproduction I'll die, or if you won't do reproduction you will die? Not like that, right? It is not essential for the survival of individual. It is only essential for the continuity of the species. Is it a defining feature or a characteristic feature? It is a characteristic feature. But wait, what is characteristic feature or what is defining feature? Defining feature is nothing but the feature which is seen in all living organism without any exception. But when we talk about reproduction, it is seen in the living organism, but exceptions are there. For example, your mule, for example, your infertile couple, right? Now, due to huge biodiversity, see, we know that we have already studied, we come under the 12 mega diversity, right? So we have huge amount of biodiversity. So where huge amount of biodiversity is present, each organism has its own mechanism of reproduction because they have their different habitat, they have their different internal physiology. So they will be having different mechanism of reproduction. Factors to decide mode of reproductions are your habitat, for example, aquatic if we'll talk about the amphibian, for example, frog. Can frog reproduce on terrestrial land? No, it has to go back in the water to lay sperm and egg so that external fertilization can take place, right? But what about the terrestrial animals? What about us? We go with the internal fertilization. By this, am I saying that all terrestrial animal goes for internal fertilization? No, right? Now, internal physiology of organism. For example, if you must have heard about hen, there will be internal fertilization in hen. Internal fertilization in hen, when I am saying that sperm will enter into the body of the female hen and fertilization will occur in the body, and after that it is going to lay eggs. So fertilization is internal, but body cannot support the birth of the baby, okay, the development of the embryo. That's why they are supposed to lay egg, right? So depending on the internal physiology and habitat, we have different modes of reproduction. Now let's differentiate between asexual and sexual. Asexual means it is uniparental. Uniparental means only from one parent. But bisexual, when we'll talk, it can be biparental as well as uniparental. Biparental means it is a unisexual flower. Unisexual means it is dioecious. Okay, dioecious. Only one sex is present, that is your male gamete or female gamete, right? But when we talk about the uniparental, they are bisexual or monoecious. That means both male and female reproductive cells are present in a single organism. What about gametogenesis? It is going to be absent or it can be present in asexual reproduction. But in sexual reproduction, it will be present always. Now, this is the important difference between asexual and sexual, that is gamete fusion, or which is also called as fertilization. Now, fertilization is absent in asexual reproduction and it is present in sexual reproduction. What about recombination? Recombination will occur during crossing over. Now, this crossing over will takes place during meiosis, okay, not mitosis, meiosis, right? Now that is absent in asexual because we do not have two parents. When two parents are not there, obviously crossing over won't be taking place, right? Sexual reproduction, it is present in sexual reproduction. Variation, there is no variation in asexual reproduction because we have only single parents, so identical copies are produced. These identical copies are known as clones, right? Now in sexual reproduction, variation is present. Duration of asexual sexual reproduction is less. For example, bacteria, if you'll talk, it will give 2 daughter cell in 20 minutes, right? But about sexual reproduction, it takes more time. For example, if you'll talk about the human, it will take 9 months. For elephant, nearly it will take 2 years, right? Now, asexual reproduction is simple, but sexual reproduction is elaborate and complex. Speed of asexual reproduction is fast. As I told you, 20 minutes mein khatam. Reproduction is done, right? You will get a new baby, new daughter cell. But here, sexual reproduction is slow. It takes time, okay, to produce the new daughter cell. Now, by this, we are going to start asexual reproduction. We have already studied what is asexual reproduction. What is the definition? When an offspring produced by how many parents? Single parent. Just now we studied uniparental, right? Uniparental. Without involvement of gamete formation, this type of reproduction, we call it as asexual reproduction. So, there is no involvement of gamete formation and it takes place from one parent. A single individual, that is uniparental, is capable of producing the offspring. We do not require two individuals. As a result, the offsprings are identical to one another. Now see here, for example, this is the bacteria and it is going to produce 2 daughter cells. Now these 2 daughter cells are identical to each other. A and B are identical to each other, as well as A and B are identical to the parent cell. Okay, both all are identical because only single parent is involved in this. The offspring shows uniparental inheritance. What is the meaning of inheritance? We have already discussed. IP. I told you the trick, IP, right? I for inheritance, P for process. So it is a process of transfer of character from parent to offspring, from parent to offspring, right? And since it is happening only from one parent, we call it as uniparental inheritance. Uniparental inheritance. Such morphologically and genetically similar individuals are known as clone. Okay, so when you see daughter cell over here, these daughter cells morphologically, what is the meaning of morphologically? External appearance, right? Genetically means you know about gene I am talking. So these clones are genetically as well as morphologically, these are similar. So what are clones? Organisms which are morphologically and genetically similar, we call them clones, right? Asexual reproduction is common in where you will see in protista. Protista is your unicellular eukaryote. Bacteria is your unicellular prokaryote. Archaea is your unicellular eukaryote. Then multicellular, obviously you are going to put it under eukaryote, right? Multicellular organism with relative simple organization. We are also multicellular. So are we going to show asexual reproduction? No. Please do not say that, ma'am, the cell is dividing in our body, that is asexual reproduction, because from one cell we are getting 2 cells. That is growth. That is not reproduction. What did I tell you before in the first chapter? That only in unicellular organism growth and reproduction is synonym. But when we talk about the multicellular organism, growth and reproduction is not synonym, right? Now, the methods of asexual reproduction that occur in many protozoans. So, 2 types of asexual reproduction you will see in protozoan. One is your binary fission and second one is your multiple fission. When we talk about the binary fission, the most common method is your binary fission. Okay, so for the asexual reproduction in protozoan, the most common method is your binary fission. When it occurs only and only during the favorable condition. Now, what is the meaning of favorable condition? It is nothing but the perfect environment, temperature, climate, light, food, nutrition. All these are when favorable, then only binary fission occurs. How many offsprings are produced? Bi means 2. Fission means breaking. So while dividing the parent, you are going to get 2 daughter cells, right? Now what is the definition? Division of the parent cell into 2 daughter individual, we call it as binary fission. Now how this, I told you what is binary fission, that is division of parent cell into daughter cell, but how it happens, what steps are involved, let's see that. So it is going to involve karyokinesis and cytokinesis. Okay, karyokinesis and cytokinesis. Karyokinesis is nothing but, karyo means we have already studied, it is nucleus. So division of nucleus we call it as karyokinesis. Cyto means cytoplasm. Division of cytoplasm, we call it as cytokinesis. Please do not write cytokinesis first, because always your karyokinesis, your brain has to multiply first, right? Then other body is required. Just imagine a body without brain, like we have in our class Himang. Body without brain, no brain at all, lost somewhere else, है ना? So karyokinesis is really important. So before first, so first it's going to be karyokinesis followed by your cytokinesis. Now, the plane of fission differ in different protozoan based on the axis of cytokinesis. The binary fission is classified into two. One is your longitudinal as well as another one is your transverse. Now, see here, this is one organism and this is second organism. If I divide this organism like this in a longitudinal way, we call it as longitudinal binary fission. If I am going to divide organism like this in a transverse way, we call it as transverse binary fission. So here also two parts, two daughter cells are produced. Here also two daughter cells are going to be produced, right? Now let's jump on longitudinal binary fission. Longitudinal binary fission basically you will see in flagellates. Whom you will see? Flagellates. For example, your Euglena and Trypanosoma. Axis of cytokinesis. What is the plane onto which cytoplasm divides? Cytoplasm will divide longitudinally. Cytoplasm will divide longitudinally. What is the mechanism? Mechanism is nothing but the body divides into two equal halves. How it is going to divide? Longitudinally, right? Now this is super important. It is a neat question. Karyokinesis is followed by cytokinesis. It is important to understand followed by whom. So before nucleus division will occur, and after that cytokinesis, that is cytoplasm division occur. Now what is the division which is followed in longitudinal binary fission? So we are going to follow symmetrogenic division. When I say symmetrogenic means symmetry I am talking. Do you remember we studied symmetry? Suppose if this is an organism. If this is an organism, okay. Now if I want to get two equal halves, so I am going to cut through this plane, right? Two equal half I am getting. So both 1 and 2 are symmetry to each other, right? So same way, symmetrogenic is nothing but from one daughter euglena, from one organism you get two organisms, and these both daughter cells are mirror image to each other. They are exactly similar to each other. This kind of division we call it as symmetrogenic division. Now, before moving ahead with the longitudinal binary fission, it is important for you to understand the structure of euglena. So this here you can see this outer layer which I am drawing now. This outer layer is nothing but it is your pellicle. It is your pellicle. What is pellicle? Pellicle is nothing but it is a proteinaceous layer which is flexible and which allows cell to change its shape as and when required. So this is your pellicle. Now another one which you can see here, let me change. White color we have. See, this white color one we have cytostome. This white color one is cytostome which acts like a mouth. Then we have a basal body over here. Basal body is present over here. From basal body what will originate? From basal body your flagellum will originate. Your flagellum will originate. One is long flagella, other one is the short flagella. Right? Then we have this kind of structure which we call it as paraflagellar body. But before that you have to understand this structure. Let me draw with this color. This one, this is just below the cytostome. Just below the cytostome it is cytopharynx. So cytostome acts like a mouth. Below that we have a muscular tube like structure. We call it as cytopharynx. And then we have paraflagellar body. And then we have basal body which is also called as blepharoplast. And in between the space which you can see here, these are nothing but the reservoir which is going to reserve the components for regrowth of the euglena. Then here this white color one you can see here, this is the contractile vacuole. Okay, what is the function of contractile vacuole? It is going to remove the excess amount of water present in euglena. Now euglena stay in water, so obviously through plasma membrane water will enter inside the body. So if excess water is there, so what will happen? Euglena will swell and burst. Euglena doesn't want to die. So what will happen? Contractile vacuole will take all the excess water. Once it is filled, it will come near the plasma membrane and it will expel all the water, right? Then here red spot which you can see, that is nothing but your eye spot or stigma. And these lines which you can see here, these are the myonemes, contractile fiber. Then we have cytoplasm, we have one nucleus. Around the nucleus we have chromatophore. Chromatophore are nothing but the specialized structure which helps in photosynthesis. Okay, then this yellow color one which you can see, these are nothing but the paramylum. Paramylum is nothing but the stored carbohydrate food. For example, in animals stored carbohydrate name is, what is the form of stored carbohydrate? It is glycogen in animal, and in plant it is starch. So if I talk about Euglena, it is paramylum in case of Euglena, right? Now let's see this. You don't have to remember what is it and function. You have to just remember its parts and where it is present. Okay. Rest of the things are only for your understanding. So flagella, it is a whip-like structure where it is present on the anterior side. Helps in locomotion. Which one? Long one. And short one will help in ingestion of the food. It will bring the food. Okay. Now cytostome is a mouth-like structure which is also present anteriorly. And what do we do with our mouth? Ingest food. Right? Cytopharynx is a muscular tube, again anterior side, and it helps in the engulf of the food particles. The way you engulf, same way Euglena also engulf, right? Paraflagellar body and photoreceptor and stigma and eye spot both are light sensitive. One is the region, other one is organelle. Both are present anteriorly and both will help in photosynthesis. Okay. Photosynthesis in the sense it will help in detection of the light. Light is necessary for photosynthesis. So in order to do photosynthesis, it will start detecting the light. Reservoir is a storage site which stores the component for regrowth of Euglena part. Contractile vacuole is an organelle which remove excess water. This we call it as osmoregulation. Osmoregulation is nothing but maintaining the balance of water and salt. Blepharoplast is a basal granule and it is present anteriorly and it helps in the formation of flagella. Chromatophore is a ribbon-like structure over here, but you can think that it is a specialized chloroplast and it helps in the photosynthesis. Where it is present? It is present around the nucleus. Paramylum is the stored form of carbohydrate. Again, many glucose will combine together to form paramylum in Euglena, right? Myoneme is a contractile fiber which helps in locomotion. Where it is present? Beneath the pellicle. So your pellicle is over here and just beneath the pellicle you will have myoneme. Nucleus is present in the center and contains genetic material. Pellicle is a flexible proteinaceous outer covering which protects and helps cell to change its shape. Now cytoplasm, obviously it helps in the biochemical reaction, right? Now, let's see the mechanism of longitudinal binary fission in Euglena. Under the favorable condition, cell grows to its maximum size. We have seen the sequence in cell division, that is your G1, S, G2 and M phase, right? G1, S, G2 and M phase. So these whole sequence, when I talk about G1 and G2, these are the growth phase. So what is growth phase? All the protein, RNA, all the components which are required for duplication, which are required for the reproduction, is going to duplicate over here. Now when the number of component increases, obviously the cell size also increases. So once it grows maximum, it stops feeding. Once it stops feeding, what will happen? Your nucleus, important component, obviously brain of the cell, nucleus, then basal granule. If it is not there, flagella won't be formed. Flagella formation is not there, there won't be locomotion, right? So basal granule, chromatophore, that is, it helps in photosynthesis. No photosynthesis, it will starve and die. And then your cytoplasm, no biochemical reaction occur. No biochemical reaction occur means automatically again you will die. So all these four components which are important are going to divide through mitosis. What is mitosis? It is a cell division in which equal number, it is a cell division which is equational. When I say equational, it means the chromosome number which is present in the parent, same chromosome number is being distributed in the daughter cell. If it is diploid, daughter cell will be diploid. If it is haploid, daughter cell will be haploid, right? Now so this process of division of nucleus, we call it as karyokinesis. But what are the other components which are not going to divide? Stigma, paraflagellar body, and contractile vacuole does not undergo mitosis. So if it does not undergo cell division, obviously it is going to disappear. Just imagine your cells, your skin cells are dying every day. Now if it does not undergo mitosis, does not undergo cell division, what will happen? There will be a point where you won't see skin, only bones will be visible for you, right? So that's how it is important. If it is not going through mitosis, obviously it will disappear. Now this karyokinesis result into two daughter nuclei. After that, cytokinesis start. How division of cytoplasm takes place? It occurs longitudinally. Groove means khadda hota hai. So this is your groove. This is your groove. Okay. Groove develops which side? Anterior side. आगे की side पर होता है, पीछे की side पे नहीं होता है, anterior side. These grooves deepen. This grooves deepen towards the posterior side. This is your posterior and this is your anterior. Then two daughter cells are formed. Now out of these two daughter cells, one daughter euglena will retain parental flagella. Other daughter individual develops new flagella formed from newly formed basal granule. So one will have parent flagella but other has to form its new flagella. What about the parts of euglena which will disappear due to no mitosis? They are going to reappear because these are important for the euglena, right? So stigma, paraflagellar and contractile vacuole are going to develop freshly in both daughter euglena, right? Now here you can see the picture. So this one, so anyways, it's showing two division of the flagella. But what is given in your textbook, it is like one daughter will have flagella, another will have after cell division, right? So this is the diagram given in your textbook. Class, tell me what is the difference between chromatin and chromosome? What is the difference? Both are the genetic material. Chromatin you will see before cell division and it is less condensed. It is less condensed. But when we talk about the chromosome, you will see during cell division and it is more condensed or highly condensed. Okay, highly condensed. That's how you are able to see them, right? So here your chromatin, which is when the cells start duplicating, it forms chromosome. Then your longitudinal grooves are formed and then you have first division of the nucleus will occur. So once the division of nucleus occur, longitudinal groove will occur and there you have two euglenae, right? So that's all we have in today's class for longitudinal binary fission. Transverse binary fission we will study in next class. Thank you.